I don't know about you, but I've had an awesome week. Granted, I was in the lab a lot longer than I expected to be earlier on in the week. It was good. My week was good because I got awesome results. I like how you can see myself in my glasses. A little trippy. I kind of look like a bug with these sunglasses, but I'm not taking them off. No, my eyes are not bloodshot. So yeah, awesome results in lab this week. It looks like my bacteria grew my plasmids. So last week I had taken 2 different plasmids and cut them into a big piece from one of them and a small piece from the other. And then earlier this week I combined the small piece and the big piece to make one new plasmid. And it looks like my bacteria took them and grew them, which is awesome! I still have to do a couple more experiments to, verify for sure that's what it is, but it looks like it's all downhill from here, which is awesome. So after I verify what is what, I will grow more bacteria in a liquid culture, and then basically I'm going to steal their plasmids that they've been growing for me, and it's going to be awesome. So I did a ligation reaction, which means I took this big piece of DNA and a small piece of DNA, and I put them in a test tube with an enzyme called ligase, and it connected them together. So it yielded me what I hoped to be one circular plasmid containing both parts that I wanted. That was a reaction I let sit overnight to make sure it was good to go. I then took that reaction, took a little bit of it, and I threw it into a little tube of cells, which are made to take on plasmids. I let them sit on ice for a little bit, gave them a false sense of security, and threw them in a water bath at 42 degrees centigrade. Which isn't like that hot, but the bacteria are like, oh crap, what is this? Why am I on fire? This hurts! And after they have that tortured for 30 seconds, I throw them back on ice, and they're like, oh my gosh, I'm panicking. I am so panicking. I will totally take on any DNA that's in this little environment that you have put me in. So basically, I torture my bacteria until they take on my plasmids. And then, I just, let them grow overnight and see if that works. I'm gonna give you a hint, it looked like it worked. Because I tortured my cells, I thought I'd be nice and let them grow for an hour before I played with them more. Here, I'm going to take my cells from my little tube and put them on 3 different plates at 3 different volumes. Just so when they do grow, they're not covering the entire plate because I want to be able to take single colonies for testing. Ooh, fire! Now, on these plates is some antibiotic ampicillin, which will kill all bacteria, unless they have a resistance for it. There's a gene called AMP-R, or ampicillin resistance, and we put that gene on the plasmid. So, if any of the bacteria took up the plasmid, they will be able to grow on the plate because they are not necessarily affected by ampicillin. So it's a nice way of telling if they have my plasmid or not. So, after plating my cells, I put them back in the 37°C room to let them grow overnight. And when I checked them in the morning, they had grew for the first time in a long time, so it looked like a success. And then here you can see my bacteria cells on my plate. They're the small little white specks in the middle of it. It's a little hard to see, but they're there. So then I took 10 of those bacteria cells from that one plate and I put them in a PCR reaction with these primers. Basically what a PCR reaction does is make exponential copies of a piece of DNA. A piece of DNA is designated by which primers you use. So specific primers will bind to specific spots on the DNA. I used 2 primers which were supposed to bind onto my very, very small piece of DNA. So after the PCR reaction was done, all we had to do was run the 10 samples on a gel and look for that small little piece of DNA on there again. And if it worked, that would insinuate, maybe more mean that my insert was there. So the piece of DNA I wanted in my plasmid was there. And I put my gel in the UV box and bam, they're all there. It looks beautiful. So that's that really. I'm going to post another short video right here of me making my plates to grow my bacteria on, just because it looks kind of cool when sped up. And I will keep my channel updated with how my project's going. I hope to have something really, almost finalized by this week before I go home next weekend for a long Columbus Day weekend. Anywho, thanks for watching.